The overall goal of the following experiment is to generate oligodendrocyte precursors from embryonic stem cells via a series of culturing steps over 30 days. This is achieved by first generating embryoid bodies from mouse ES cells to initiate cell differentiation. As a second step, the embryoid bodies are treated with the small molecules retinoic acid and pure morphamine, which induce the directed differentiation of the cells into oligodendrocyte precursors. As a third step, embryoid bodies are disaggregated into single cells. After an additional 22 days in culture, approximately 80% of the cells are OPCs. Next, the MESC-derived OPCs are transduced with baculoviruses carrying NAV1.2 subunit in order to generate spiking OPCs. Results are obtained that show the expression of OPC marker and spiking properties of these MESC-derived OPCs based on immunofluorescence microscopy and electrophysiological recordings.
Hi, I'm Peng Jiang from the laboratory of Dr. Wenbing Deng in the Department of Cell Biology and Human Anatomy at the University of California, Davis. We are also affiliated with the Shriners Hospital for Children of Northern California.
Hi, I'm Vimal Selvaraj, also from the Deng Lab.
Today we will show you a procedure for differentiation of mouse embryonic stem cells into oligodendrocyte precursor cells.
We use this procedure in our laboratory to study the properties of embryonic stem cell-derived oligodendrocyte precursors in order to generate cells with better therapeutic potential.
So let's get started.
Today, oligodendrocyte precursor cells, or OPCs, will be generated from the OLIG2 GFP mouse embryonic stem cell line. These embryonic stem cells need to be passaged every 3 days to keep them healthy, at an optimum density, and to prevent spontaneous differentiation. Passage the ES cells when they are at 70% confluence. Under a laminar flow hood, remove the medium over the culture, rinse with PBS, and trypsinize with 2 ml of pre-warmed 1X-Trip-LE Express for 5 minutes at 37 degrees Celsius. Stop the reaction and resuspend the cells in 4 ml of MES cell medium. Transfer the cells to a 15 ml conical tube and pellet them at 1000 RPM for 5 minutes. Resuspend in MES cell medium. Plate 1/10 of the cells over a confluent layer of irradiated MEF feeders in MES cell medium. After 3 days, ES cells are approximately 70% confluent and are ready to be passaged or suspended to form embryoid bodies. To induce embryoid bodies and begin differentiation, trypsinize mouse ES cell colonies as before, and then resuspend the cells in knockout serum replacement, or KSR, medium. Count the cells on a hemocytometer. Dilute the suspension in KSR medium to 15,000 cells per milliliter. Plate 3 ml into each well of an ultra-low attachment 6-well plate. Culture the suspended cells at 37 degrees Celsius for 4 days. Change the medium completely every 2 days. Starting on day 4, replace the medium daily. On the 4th day, supplement the KSR with 0.2 micromolar retinoic acid. On the 5th day, supplement with both retinoic acid and 1 micromolar pure morphamine. On the 6th and 7th days, feed the embryoid bodies with N2 medium supplemented with 0.2 micromolar retinoic acid and 1 micromolar pure morphamine. After 8 days in culture, the majority of embryoid bodies express GFP and are ready to be disaggregated. Trypsinize and count the cells as before, then resuspend them at 3.5 times 10 to the 5th cells per milliliter in OPC medium. Replate 3 ml of the suspension in OPC medium using polyornithine-coated 60 mm dishes. Approximately every week, the cells will become confluent and will look like this. Passage the cells as before with a 3-minute trypsinization. 30 days after the original resuspension in KSR medium, the cells show characteristics of oligodendrocyte precursor cells. To generate spiking OPCs, use mouse ES cell-derived OPCs that are between 50 and 70% confluent. Exchange the medium for 1x PBS to rinse the cells. To introduce voltage-gated sodium channel 1.2 alpha subunit, we use the BACMAM sodium channel kit. Mix 1 ml of BACMAM component A with 4 ml of 1x PBS. Remove the PBS rinse, and add the diluted component A. Incubate at 37 degrees Celsius for 2 hours. Replace the BACMAM reagent with OPC medium enhanced with components B and C as described in the kit instructions. Incubate for an additional 2 hours at 37 degrees Celsius. After the incubation, replace the enhanced medium with fresh OPC medium. After another 24 hours of culture, the ES cell-derived OPCs can be assayed for spiking. At this time point, the EBs do not show any GFP fluorescence. The EBs show strong GFP expression and are ready for disaggregation. Culturing for about 30 days after original resuspension in KSR medium, the majority of cells are immunopositive for NG2 staining, which is consistent with GFP expression. However, unlike brain OPCs, these MES cell-derived OPCs fail to fire action potentials, even when the cells were depolarized to 0 millivolts. After introducing NAV 1.2 subunit by viral transduction, these MES cell-derived OPCs get excited and fire action potentials.
We have just shown you how to differentiate mouse embryonic stem cells into OPCs and generate spiking OPCs by viral transduction.
When doing this procedure, it is important to remember to store the retinoic acid, permorphamine, and FGF2 properly, and always use fresh aliquots. Discard any extra thawed retinoic acid after use.
So that's it. Thank you for watching and good luck with your experiments.